My voice is okay, I think, but anyways. So it's a pleasure to have, this is the closing colloquium, the 56th colloquium. Nobody else can do this. 56 consecutive colloquium. Göker Arpağ, one of our own, is going to give this colloquium. So Göker is a long-time friend of some of us. He got his undergraduate degree at Istanbul Technical University. The Istanbul Technical University had its golden years with some of us there, with Önder Hocam there, Sondan Hocam there. I was there during those golden years. We had many golden children who graduated, and his career afterwards proves how golden he is. He went at Worcester Polytech Institute. Worcester, that's how Americans pronounce it. I think it's written Worcester, but it's Worcester, just like Worcestershire sauce. It's Worcester Polytech Institute. So he got his PhD there, and then he did a postdoc at Vanderbilt University, and then Sondan Hocam recruited him to come to our university, just like Yasemin Hocam last time. So we are, I think, very glad for the country, for the university, and for these young scientists to be with us. Göker he's trained as a physicist. Well, it's physics engineering, but at Istanbul Technical University, if you take physical education, it's called physical education engineering. So it's just basically physics. He's trained as a physicist, but even in his undergraduate years, as all good students, he was doing research there, and his research is biological applications of physics. And it is really molecular biology, cell biology, developmental biology. And another very important thing about that, I know one other person like that, myself, is that he's trained as a theorist, but he also does experimental work. So this is fantastic to be able to put both things. I had a professor called Bill Macmillan. Some of you may have heard of him, at Illinois. He was both a theorist and an experimentalist. So it's great to have him here. Also, this is his birthday. Happy birthday. Doğum gününüz kutlu olsun.
Teşekkürler.
I will have more to say on this towards the end of the colloquium. So stay put, don't go away. Okay, the floor is yours.
Yeah, thank you for a kind introduction, Nihat Hocam. Yeah, I'm very happy to be here, actually. I joined Kadir in September, but I will talk about one of my postdoctoral work at Vanderbilt University, which we carried out in close collaboration with a PhD student. Now she also graduated and doing her postdoc, but we've been working on basically microtubule dynamics. So let me first tell you about the microtubules in general. Microtubules are polymers, and they are a member of a cytoskeleton. So cytoskeleton, you can think about it as the bones, the skeleton of the cells. As you can see here, we have many different cell morphologies, and the microtubules are basically following this morphology, basically as supporting this specific cell shape. And microtubules are involved in many cellular processes. For example, you see a motile cell here. They support for the cell migration. And also you see in this column, and there is the flagellar structure basically consisting of microtubules and some other proteins that drive this beating cilia and helping this organism to basically move forward. And microtubules are also important facilitating the intracellular transport. So here is an animation. You see that these are the microtubules, and they are like the highways, the roads in the cell, and these motor proteins actually walk on the microtubules attached to cellular cargoes such as organelles and vesicles, and the microtubules are providing a way to, as seen in this live cell imaging, microtubules are in green. They are basically a network of roads within the cell and helping transporting of cellular cargoes across different regions within the cell. And the microtubules are also important. I think the most amazing structure in the cell is during cell division is the mitotic spindle. So they form this mitotic spindle attaching to the chromosomes aligned in the middle of the cell and then separating them. After this separation, cell divides into two, generating two different cells. And in general, microtubules are very important. So because of this spindle structure, they are implicated in cancer. So most of the cancer drugs, cancer treatments, chemotherapical treatments are, some of them are targeted for the microtubule dynamics. But also in other tissues, for example, in muscles, you see the contribution of microtubules where the muscle contracts, microtubule rigid polymers are basically determining the stiffness of this tissue and affecting how successfully the contraction can happen. And also, as I showed flagella before in the clammy, but if you think about it, sperm cells are having this flagella and microtubules are the main driving factors here. So like this is this proper function of the microtubules are important for fertility in general. So if we go back to cells and we see microtubules are in red here, they are very highly dynamic, and this dynamic behavior is very important for proper functions, proper cell functioning. And here you see another protein called EB1 in green. This is recognizing the growing ends of the microtubules, and I will describe it further in more detail. But this dynamic behavior can be reconstituted in vitro also. So what I mean by in vitro is we take out protein components from cells and then put them on the microscope and see. And here is a microtubule growing from centrosomes. So centrosomes are microtubule organizing centers. Well, microtubules originate, nucleate, and grow out of it. And as you would appreciate that this dynamic behavior is very reminiscent and can be reconstituted in vitro. And if you carefully look at it, microtubules polymerize, basically grow, I will refer to that grow, and then stop growing at one point and then start depolymerizing again, which I will refer to as shrinkage. So this overall behavior, basically switching between polymerization and depolymerization, is termed a dynamic instability, and which is the basically hallmark behavior of microtubule dynamics. So if I tell you a little bit about the details of the microtubule itself, they are consisting of tubulin heterodimers, which has basically two monomers, alpha and beta, but it's hard to separate them. So this is the subunit for this polymer. And then they are attaching to the polymer in a head-to-tail fashion. So there is structural differences between the two microtubule ends. And in the microtubule field, we name them as plus N and minus N. So these tubulin dimers get together, forming actually individual filaments, but about 13 of them is forming the microtubule itself. And this is actually a cylinder, hollow cylinder, empty inside, and 25 nanometer in the diameter. So if we look at this structural study of the tubulin dimer, we see that there is GTP. It's a nucleotide providing the energy. One of the energy sources in cells is attached to this tubulin heterodimer. But in the beta subunit, this GTP then hydrolyzes into GDP. That's why the microtubule gets its energy to self-assemble. And that's why we can call tubulin a GTPase. Throughout the talk, I will mention this nucleotide state of the microtubule, which I will refer to the nucleotide found within the beta subunit here. So if we try to look at the microtubule dynamics, so we have a pool of GTP tubulin that is being added to the polymer, as I mentioned, in a head-to-tail fashion, and the polymer grows basically. And then this hydrolysis happens after a lag. So after some time, and the lattice, what we call, is now consisting of GDP-bound tubulin, which is actually structurally different from the tip of the microtubule. So we can actually now divide the polymer into two regions where there is a cap of GTP-bound tubulin. So I will just, in short, tell this GTP cap. So we can actually relate this cap size to the microtubule growth rate and this hydrolysis rate. So the ratios of these two parameters will approximate the GTP cap size basically. So this is the standard model in the field. So I will refer to this as the GTP cap model. So if you grow slowly, you will eventually have smaller GTP caps. And if you grow faster, you will end up seeing a larger GTP cap. So this GTP cap is actually explaining the dynamic instability. So if you lose this cap, because of this structural differences in the lattice after hydrolysis, the tubulin is now unstable. And when this GDP lattice is exposed, microtubule stops polymerizing and going through an event called catastrophe, and it starts depolymerizing. And the opposite event can happen, so the shrinkage will pause and microtubule will start growing again. But I'm not going to talk much about the rescue part. I will more focus on the relationship of microtubule growth and the catastrophe. So the idea here is that the longer GTP cap you have, it is harder to lose the cap because the tubulin subunit addition to polymer is stochastic basically. And the smaller caps are more likely to lose, so you would expect to see a more frequent catastrophe. So if we come back here, so we can assume a constant growth rate, constant GTP hydrolysis rate, sorry. And if the growth rate is small, there will be smaller GTP caps, and it is more likely to expose this GDP lattice, so we will observe more catastrophe events. And for the opposite, if microtubule grow fast, the cap will be larger, and there will be infrequent catastrophes. So this is the standard model since the discovery of dynamic instability 40 years ago that is well accepted in the field. But if we assume...
Can I ask you a question? What determines the GTP hydrolysis rate? Because it seems that the stability of the microtubule depends on this hydrolysis rate. If it is fast, and then it's going to be more stable, I suppose. This is what I understood.
Yes, if the hydrolysis rate is fast, since this is in the denominator, the cap will be smaller. Okay. Actually, the units or the rate is 1/2. So that's why, like, fast is the opposite way.
What determines the hydrolysis rate?
So it's measured in 1/2, so not second.
What factor make this rate faster or slower?
So actually, that's what I will talk a little bit about it today. But yeah, I will mention that later in the end. Yeah. But if we would be assuming constant growth rate, we can basically, with smaller, slower hydrolysis rate, will generate larger GTP caps infrequent catastrophes and the opposite, basically. So now, based on this GTP cap model, we have two questions arising. Do slow-growing microtubules have smaller caps and frequent catastrophes, and also the opposite? Fast-growing microtubules should have larger caps and frequent catastrophes. So to understand this relationship, as I mentioned, we use an in vitro approach. So we purify proteins from cells, in this case tubulin, and then we are using a nucleotide called GMP-CPP, which is an analog for the GTP I mentioned, but the hydrolysis rate here is very, very slow. So that's why this microtubule becomes stable. It doesn't depolymerize, at least during our experimental time, which is about like less than an hour, actually. And then we attach these microtubule seeds, as we call them, using antibodies to glass cover slip. And then we put them on the microscope. We specifically use total internal reflection fluorescence microscopy. The idea there is to send a laser with a given angle so that it will bounce back totally, but this creates an evanescent field which is just illuminating very near surface on the glass substrate, and then basically we are having increased signal to noise ratio. And then when we add purified tubulin, we can basically generate microtubule dynamics, as you see in this movie. To quantify it, what we do is draw lines on these movies and then basically generate a time versus like position plot, sort of. So this is basically showing the length of the polymer over time, and as you would appreciate that the polymer is lengthening and then polymerization stops. This is a catastrophe event and shrinks back. So we can measure the growth rate and also count the number of catastrophes and calculate the catastrophe frequency. So now this is how we can characterize the microtubule dynamics, and we need to find a way to measure the size of the GTP cap. So remember the protein I showed earlier, which was in green in the movie, called EB1, and this is actually going on the growing tips of the microtubules, and it is actually autonomously tracking the growing microtubule ends, and it can also partner with other proteins in the cell and bring them onto the microtubules. And how it does is basically recognizing this nucleotide state. So it is basically seeing the GTP on the microtubules and then localizing on the growing ends. And after hydrolysis, it doesn't localize there, as you see. So here is another movie for the microtubules, and this EB in green. So as you would appreciate, this EB is looking like comets. Especially it is seen in this chymograph. So to quantify it, we can again measure this intensity for a given time point, for example. And the hydrolysis is accepted as a stochastic process. So it's like a radioactive decay. So over time we will lose a number of GTP-bound tubulin subunits over time, so it is expected to be an exponential decay. But since we are doing fluorescent imaging, so there is something called a point spread function. That's why we don't see a pure exponential, but a convolved image. I'm happy to talk about it later on. I don't want to go so much into technical details. Actually, one of the work I'm continuing is basically better determination of this GTP cap size. So coming back here, we can, for example, try to establish a relationship between the growth rate and the catastrophe frequency. So one way of increasing the growth rate is adding more subunits. As we increase the concentration of subunits, microtubule will grow faster, basically. But when we measure here, like the catastrophe frequency, we see an inverse relationship. So at lower concentrations, microtubule grows slower, many catastrophe events, but as we increase the concentration, there is virtually no catastrophes, and microtubule is growing fast. And when we measure the comet size, the GTP cap size, basically, and we see a linear relationship between the growth rate and the GTP cap size, as we increase the growth rate, the cap size is increasing as expected. So these questions, do slow-growing microtubules have small GTP caps? Yes, and they catastrophe frequently. And fast-growing microtubules have larger caps, and they catastrophe less frequently. So I want to remind you that this is in vitro. We are using purified tubulin, but I also want to remind you that microtubule is having actually two ends. So these measurements I showed you was for plus n, but there is also minus n. So minus ends of the microtubules were not studied very much, even though like it is appreciated that there are two ends of the microtubules. That's why even in like earlier times, it hasn't been changed in the past 25 years, that the minus ends are considered to be dark side of the microtubule dynamics. But as you would appreciate, that the dynamic behavior is a little bit different than the plus end. So they grow with like slower rates, and they also go through catastrophes, and this dynamic behavior is independent of each other. So if we want to establish again the growth rate and catastrophe frequency relationship, we see that the minus ends are also decrease in the catastrophe frequency as we increase the growth rate. But for a given growth rate, for plus ends and the minus ends, we see that actually minus ends are catastrophing less frequently compared to the plus ends. So coming back to this GTP cap model, we would basically assume that the minus ends will have inherently larger GTP caps because they go through less frequent catastrophes at a given growth rate. So when we look at this minus ends and EB1 localization, we actually see that the scaling between the growth rate is actually the same. But when we are comparing the same like growth rates, there is no difference actually with plus and minus ends. But overall, slower growing microtubule minus ends are having smaller EB comets. So our hypothesis is not true in this case. So like I will actually like skip this like a little bit fast, but the reason for this stability is what we found is because of the off rate of the subunits, but I will be happy to discuss details, but it will be out of the subject. But what I want to say with this work is that the GTP cap size is not the sole determinant of microtubule lifetime. Lifetime here, I mean that how long microtubule stays in the polymerization phase. So it is inversely related to the catastrophe frequency. So like coming back to these questions, So we have these true for plus ends and within minus ends itself, so the relationship was still valid there. But in general, we can already tell that the GTP cap is not only determining the catastrophes. So what we can do is now try to change the catastrophe rate instead of changing the growth rate and see what will happen to GTP cap size. So as we would appreciate that the cells have many different proteins, and some of these proteins, actually many proteins, regulate the microtubule dynamics. So these are called microtubule associated proteins. So one protein called clasp is actually stabilizing the microtubule dynamics. So let me tell a little bit about this protein. So like it is basically found everywhere in different stages of cell cycle. At interface, it's at the cell cortex. In mitosis, it's at the equator. It is involved in cell migration. It is involved in neuronal growth. And what it's doing is, as I said, stabilizing microtubules basically through suppressing catastrophes, as we would see here from like another work from the other postdoc in the Zanik lab. So when clasp is added, we see that the number of the catastrophe frequencies are decreased and in the presence of clasp. But what's different here is that the growth rate actually doesn't change when we characterize the growth rate. So coming back to this GTP cap model, we can then hypothesize that clasp is slowing down GTP hydrolysis rate and then increasing the GTP cap size. So actually, when we look at the EB localization, we see that there is no difference between control and the clasp condition. So the comet localization is basically the same, and we can conclude that the clasp does not increase EB comet size. So what it's doing is basically we try to understand what's the reason for this. We looked at the microtubule growth at high temporal resolution. So this is an average growth rate, so we can do a linear fit. And these residuals, basically the distance to this average curve, we can try to use this as an information of how straight the growth is or how wobbly the growth is. And what we found is that in the presence of clasp, microtubule is basically growing in a more wobbly fashion, deviating away from this average length. And our actually collaborators also showed that the reason for this wobbliness is because clasp is preventing microtubules to go through catastrophe. And it switches back to the growth phase. That's why there is this wobbliness is coming from. However, we still don't know the specific mechanisms underlying how clasp is affecting the microtubule dynamics. But what I can tell you is that this is another evidence we have that the GTP cap size is not the sole determinant of microtubule stability. So coming back here, these are still valid observations, keeping in mind that the cap size is not the only way to determine the stability of microtubules. Now, we go back to these cells. What I want to tell you is that the dynamic behavior in cells is a little bit different than what we are seeing in vitro because of the many proteins involved there. So the growth rate in cells are an order of magnitude higher compared to what we are measuring in vitro, yet the microtubules are going through frequent catastrophes. So they are are highly dynamic in cells. So how this is possible based on this GTP cap model that we have? So what we can do now is try to increase the growth rate, and using a protein called XMAP215, which is a polymerase. So it's found in the mitotic spindle, and it is actually speeding up the growth rate an order of magnitude. And it's acting as a polymerase. It's basically actively adding tubulin dimers to the growing microtubule ends. And since we are using EB1 already to determine the cap size, I also want to tell you that these two proteins synergistically increasing the growth rate even further. So when we look at the addition of XMAP215, we can appreciate that the more XMAP215 we add, the faster the growth rate we can have. And we can also quantify the catastrophe frequencies. As you would appreciate, there are like 3 catastrophes here without XMAP215, and there are more catastrophes in the presence of it. But we were expecting to see less catastrophes. If anything, XMAP215 is actually making microtubules to go more frequent catastrophes, even though they grow at higher rates. So this relationship I showed you earlier with tubulin alone is the expected behavior with the, based on the GTP cap model, but XMAP215 is breaking this relationship basically. And we can again compare the growth rates at the same growth rates with and without XMAP215, and catastrophe frequency is actually higher because we had virtually no catastrophes without the XMAP. Now the question is how this protein is able to promote catastrophes instead of, in spite of making them grow faster. Now we can hypothesize that this is decreasing the GTP cap size, so therefore catastrophes are promoted more. So like I can bring this back. So if the GTP cap size is decreased, the way to do it is increasing the hydrolysis rate. So when we look at the EB localization, we actually see that the cap size is not decreased actually. If anything, EB localizes even more on these microtubules that are growing at the same rate with the presence of XMAP215. So this is another evidence that the GTP cap size is not the determinant of the microtubule lifetime. So our hypothesis is wrong in this case. So now we can come up with an alternative hypothesis telling that XMAP215 will basically perturb how the microtubules are growing. And we can again look at this deviations from the average growth rate and see that the XMAP215 is actually providing microtubules to grow more wobbly in that sense. But further, what we observe is that the microtubule ends are now structurally behaving differently when they are growing. As you would appreciate here, we are looking at EB1 localization specifically here. So we have normal comets in the beginning, and then these comets are split into two. Remember that the microtubule is like a cylinder consisting of many protofilaments. So like the end structure is already different here, and it's even curling out a little bit from the main polymer lattice here. So what we can now tell is that XMAP215 is actually altering how the microtubule is growing changing the end structure and allowing it to have more fluctuations in the growth rate and basically disturbing the microtubule growth microtubule growth in more structural way. So what we can do now is look at the moment of catastrophe and how EB1 localizes at the microtubule end. So what we know is actually that so this is the moment of catastrophe, time 0, and the microtubule starts depolymerizing. And like within a very short amount of time, the microtubule growth rate actually slows down, basically in the last 2 seconds or so. And actually, EB1 signal is decreasing. So this is a pre-catastrophe state suggested by observed by another lab earlier. We are basically confirming that prior to catastrophe, microtubule slows down and then EB is lost. So that's why the GTP cap is eroding over time, because there is no growth happening still. What they suggested is that there is a minimum number of GTP tubulin should be there, so microtubule will not go through catastrophe. But when we look at in the presence of XMAP215, what we see that there is no slowdown prior to catastrophe, so microtubules catastrophe more abruptly, and the EB intensity is actually higher at the microtubule ends right before catastrophe. So this is suggesting that in this case we still have a GTP cap, but microtubules still catastrophe, go through catastrophe. So with that, we actually try to come up with some models. So what we started, what we can start with is low tubulin concentration. So we have slow growth and smaller GTP caps, and prior to catastrophe, microtubule slows down. Hydrolysis is catching up, we are losing the GTP cap and then going through catastrophe. At high tubulin concentrations, microtubule grows fast, GTP cap is large, there is no problem, you don't go through catastrophes. But when we have this XMAP215, microtubule growth is perturbed structurally actually. And we have, we can observe split comets or curled protofilaments coming out of the microtubule. But at the pre-catastrophe state, the slowdown is not as significant as in the case without with tubulin alone, and the growth is much more irregular, and microtubules are catastrophing with the presence of GTP cap. So what we can tell is basically the perturbations in the growth is actually leading to catastrophes, in this case, not the GTP cap size. But now the question is this the mechanism used by cells in order to facilitate fast and dynamic, fast growth and dynamic microtubule behavior? So this is where we, like Veronica actually has been working on it, even though she graduated. There is a new graduate student in the Zanik lab and continuing these experiments. But what she observed preliminary is that there is a relationship, the similar relationship in cells at different stages, interphase and mitotic stages, and it is scaling similarly with the in vitro data. So now I want to wrap up a little bit. So we need to now go beyond the GTP cap model and try to appreciate that there are other factors defining microtubule stability, and also the end structures of the microtubules can be basically different. And the one question is basically, what's the role of this GTP cap size on the microtubule stability? So we have evidence for it. We have evidences against it, especially with the altered microtubule dynamics. And how does the dynamic microtubule and structure is manifesting into the microtubule dynamics, and what are the other characteristics that the microtubule polymer will have on the dynamic behavior of them. I mean, I can't give you a short answer for these questions, because the understanding the underlying mechanism of catastrophe is an ongoing challenge for like over 4 decades. But what we know from earlier work is this catastrophe is not a single step random processes. So it's actually it's involved in many more steps. But we have recent studies on the microtubule structure, such as cryo-EM studies and also 3D cryo-electron tomography studies. And these are trying to show the details of the microtubule and structure, and the idea is how that they can be related to catastrophes. But the problem with these studies is that these are static images, so we can. It is harder to relate them to the dynamic behavior or based on the structures that is observed in still images. So that's why there are many computational models at various degrees of complexities, and there are now different visions, different ideas happening in the field, how the end structure is determining the microtubule stability actually. But what we need to remember, as I told you today, that microtubules have 2 ends basically, and we need to now think about the effect of microtubule associated proteins, because most of these studies are done in the presence of, in the absence of additional proteins. So actually, this graduate student Veronica and my advisor Maria, they just wrote a review about this GTP cap, which was published last month. So if you want to get more details, I would suggest you to read this. It would be a more extended version of my talk today. So with that, I want to thank my postdoc advisor Maria and the graduate student Veronica, and there are more members of the ZANIC lab over the years, but these are the people that were involved in the studies that I showed you today. So I can take any questions if you have.
How lucky you are! You are the only male.
Yes, yes. So I guess it was opposite in physics. So in physics there were less women, and
I usually, in physics, I am the
only women there.
Questions? I have a question.
Okay.
Now, these in the growth curves, you had a linear curve with wobbles, what you call wobbles on that. This may be completely off the point, but these kind of wobbles are I can look at them as broken sigmoidal curves. And the sigmoidal curve is the signature of a correlation. For example, in hemoglobin as opposed to myoglobin. That if one step happens and it is connected to the previous step, it's sigmoidal curves. Is this completely off line, or is there correlations down? Is there correlations down the microtubule which affect that?
Actually, I mean, it's a good point because I would expect to see some sort of correlation, even though like those individual filaments on the microtubule can grow independently, but they still need to establish lateral bonds between them to become more stable. So in that sense, if one part is sticking out, maybe it's going through catastrophes because it cannot establish lateral bonds, so it shortens back. So that's why you might see in the signal that there is an increase followed by a decrease, increase followed by a decrease. So actually, I wanted to go towards that direction, but I didn't want to go so much into like molecular dynamics part. But there are some models just to like somewhere in between, looking at the longitudinal and lateral bonds, but they don't, as far as I know, they don't go into that much detail to correlation. And actually, like in the past, we weren't able to have this high spatiotemporal imaging. So now current technology is allowing this, and I guess we can more look into these kind of details as you would suggesting. But like short answer, yes, I would expect to see some correlation, especially if the microtubule is in the growth phase, and maybe the correlation might be reduced right before the catastrophe. That's why it happens.
Thank you. Other questions?
Okay, thank you very much. It was very nice talk. So these processes are happening in a healthy cell, right? You know, degradation and the synthesis. What happens in the state of a disease case, like cancer? What's going on in the cancer cell?
So that's a good question, actually. So in the cancer cells, so they divide very fast, and there might be multiple poles, actually. Where was it? I was trying to find that image in the beginning. So there might be multiple poles, and microtubules keep dividing very fast. So that's why, as I was mentioning, like the cancer treatments might target microtubule dynamics. So that's why we want to actually understand in general how the like microtubule stability is established, and basically the way that the microtubules find those chromosomes is with several trials. It's called like there is a model search and capture. So they grow, they need to be dynamic and depolymerized because they are like shooting and seeing if the chromosomes are there. And then when they hit the chromosomes, they attach to centromeres and they become stabilized. So that's why not just like the dynamics, the growth rate of the microtubule we can change, but we can change the catastrophe frequencies if we can modulate it or understand how it's happening. So that would also affect how we would find the cancer, the chromosome. So like that could be a way to find alternative ways for cancer treatment. But you, I guess, ask more general question, what happens during cancer for the microtubule dynamics, or was that?
Yes. The other thing is that Taxol is targeting the microtubule synthesis. You know, I'm just curious to know, is Taxol stopping the degradation or formation or attacking any enzyme that synthesizes these microtubule structure?
So that's actually structurally it's binding like I guess inside the microtubule from the cylinder. It is I think affecting the mechanics of the like the physical characteristics of the polymer, because microtubules become very soft, but it's preventing basically the overall dynamic behavior. So they are also stable, but they sort of freeze the microtubule. So that's why it is used as a cancer treatment. But here, the problem with that actually, there is a Prosser lab at Penn. So he's working on the roles of microtubules in muscle tissues. So when you use Taxol as a chemotherapeutical unit so there are increased risk of heart failures because of when microtubules become softer in the muscle tissues, that changes the stiffness of the tissue. That's why they are actually bringing like problems on the beating of heart, because when you use something like Taxol chemotherapeutical units, it will affect the whole body. So the idea there is cancer cells are dividing very fast, so we can keep the dosage a little bit low, so we will see most of the effects on the highly dividing cells, but it is actually affecting the entire cells in the body. So one way that affects is in the muscle tissue.
Okay, thank you.
Yeah, there is another question there, I guess.
In the start, you said the microtubule growth rate affects different kind of microtubule growth rate affects the catastrophe rate, etc. When testing those, which methods did you use? Did you use different MAPs to change the rate of growth of the GTP, or did you use different methods to manipulate the GTP growth rate?
That's a good question. Actually relates to Sondonajam's question. I was going to come back there. So mainly we modulated the microtubule growth rate because that is more easy to do. So we use proteins, for example, XMAP215 to increase the growth rate. But also we can, in the beginning I showed, we increase the subunit concentration, so that increase the growth rate. But we also modulated the catastrophe frequency with CLASP, for example. It was making them less stable, which didn't have effect on the growth rate. But this XMAP215 was inherently bringing more catastrophes in the end. So technically, that's why I didn't want to tell in the beginning that we didn't find a way yet to change the hydrolysis rate. One problem is actually we can't directly measure the hydrolysis rate. So we are measuring the size of that GTP cap. It's also indirect measurement. And if we know the growth rate, so we are measuring the cap size, and if we know the growth rate, we can calculate the hydrolysis rate, but we can't directly measure the hydrolysis. What I'm continuing, what I couldn't finish during my postdoc is, and I'm continuing a little bit, was on basically making simulations and trying to find better ways to be able to directly measure the hydrolysis rate, even though we can still use the EB1 as a proxy for the cap size. But is this relationship is still valid, and what are our limitations? So that's what I'm exploring using simulations, because in the simulations I can control the hydrolysis rate, but there are many proteins which might be, that's another thing that I showed you only 2 proteins today. There are many other proteins, for example, statin, OP18, so that is also involved in, it's a protein, but it's involved in cancer, for example. And it is also changing the microtubule dynamics. So we need to go and evaluate the behavior of those proteins and see how they are affecting the GTP cap size. So maybe there is a protein that would be able to change the hydrolysis rate.
So more questions, but it's the top of the hour. So
okay.
We'll take more questions during. Hello. Surprise.
Kim the surprise.
O taraftan mı geliyorsunuz? Be ready, as we had talked.
Çok teşekkür ederim.
Mutlu yıllar.
Sağ olun. Teşekkürler. Sağ olun.